Today I wanted to bring you on a journey engineering swarms across scale, from the nanoscale to the robot scale. And our source of inspiration are really these flocks of birds. If you look at these birds, they can do beautiful complex dances in the skies. And there's many features that are interesting for real-world applications. For example, if you keep adding birds to the flock continues to fly, so they can scale to huge numbers. If a bird falls to the ground, the whole flock doesn't crash to the ground, so they're robust to individual failure, and together they can do more than the sum of their parts. For example, they might be better at avoiding predators. And what's fascinating is that there's no central leader telling every bird what to do. It's just the result of every bird reacting to its local environment, and that gives rise to these desired flocking behaviors. And you see examples of self-organization like this everywhere in nature, whether it's in ants as they create trails to your picnic table, or in honeybees as they make decisions about their next nest site, or the ability to grow fully functioning human beings just from a couple cells. So the challenge we face as swarm engineers is very often we have a swarm behavior we want to achieve, say flocking, decision-making, trail formation, morphogenesis, but we need to back out those individual rules, those simple designs that give you the desired behavior. And so in my team, we do this in two different ways. One is we take inspiration from the wonderful work that's been done by biologists and apply that in the engineering world. And the other, if there is no relevant bio-inspiration or there's just no known rules that we can use, then we need to explore. So that could be guessing these rule sets, or it could be doing machine learning so that we can automatically come up with solutions to our swarm challenge. So when we use bio-inspiration, we can go and put these rules on board robots. Here you see a swarm of flying robots that we did over at the Floriano lab. Here's their GPS trajectory, and here they're using the basic Reynolds flocking rules. And you see these circular topologies, these circular dances that emerge, which is actually what we predicted because we do a lot of simulation to see what should ever happen. And we throw these robots in the air, we can show that these rules work. We can also do the more learning approach. So here what you have is a swarm of robots that are learning on the go how to swarm. Their goal is to push a frisbee from the center of the arena to one side of the arena. And we're using artificial evolution of behavior trees to automatically come up with controllers that are human understandable. And here really in 15 minutes, they figure it out, from not knowing how to swarm to figuring out how to push this frisbee. And this is all done on board the robot. So we have good tools that allow us to build swarm behaviors and to push them to reality. Now, this is all interesting, but fundamentally it's, 10 robots, 20 robots, sometimes up to 100. And in my mind, swarms need to work in much, much larger numbers. And so I got excited about the field of nanomedicine and the ability of nanoparticles, because of their size, to essentially leak out of vessels into certain types of tumors. And so that makes them useful vehicles for the delivery of drugs or diagnostics directly to tumor tissue. And in Sangeeta Bhatia's laboratory, where I went from 3 years, they were able to create particles with different sizes, particles with different shapes. This might be an iron oxide nanoworm, for example. Some of these particles had different charges, or could even be energy receptive. So that meant you could use light or magnetic fields to activate them. You could decorate these particles with molecules that allow them to bind to receptors overexpressed on certain cancer cells. And you could load them with a drug that you could release in a more or less controlled fashion. So there's many knobs that you could turn on the design of these particles. And these particles, they work, per injection in the 10 to the power 13. So there's the sheer giant number that I was excited about. And so we went to think about how we could make these particles work together. But before we went into something that was a little bit too far-fetched and futuristic, we started out by scenarios that were closer to home, closer to application. So here's just one scenario. Here you have one particle. We can change its size. That changes how fast it diffuses in the tumor. And we change its coding, which changes how sticky it is to the receptors that are on our cancer cells. And in our model, these particles leak out from the vessel and need to go deep into the tumor, killing at least 20 cells deep. So they need to accumulate to the level where they kill at least 20 cells deep. And we thought that was a challenging scenario. If we could do that, we would do a decent job at the tumor we were considering. So we did loads of simulation, which is the backbone of everything that we do in swarming. And just looking at this, here you see how fast they are, here you see how sticky they are, and our goal is to kill 20 cells. And we noticed that most of these particle formulations would not kill 20 cells. And if you look on the right, these are stochastic simulations, you can basically see that just turning those two knobs completely changes the distributions of nanoparticles that you see in the tumor. For example, A was a particle that was very sticky. And so if you were to test this in a dish with cancer cells, it would look great because those particles would essentially stick to all the cancer cells and kill those cells in the dish, in those well-mixed environments. Whereas in my line of tumor cells, the particles leak out, stick to the first cells they encounter, and never go deep into the tumor tissue. So that's really an example where the collective behavior of all these particles in these complex environments does matter for the clinical outcome. And we've since then been working on making more realistic models of these tumors so that we now have cell surfaces, so that we have a better understanding of what happens if there's heterogeneity. And on the bottom, you see one of our microfluidic devices, a little tumor on a chip with blue and green nanoparticles penetrating our artificial tumors so that we can see under the microscope the things that we're implementing in silico and hopefully understand these dynamics a little bit better. Ultimately, as part of this EvoNano project, we're trying to automate the full pipeline. So say you have a tumor scenario and you're trying to treat the specific tumor. How can you use multi-scale modeling to understand where the nanoparticles go through the body, how they move through a tumor, how they move through a cell to actually have the desired impact? And how can you use machine learning to automatically design those particles so that at the end of this computational pipeline, you have a particle suggestion that you can go and test in vitro and in vivo before pushing to the more translational side in the clinic. And we also designed, as part of this process, a game called NanoDoc that allows us to explore, again, this idea of exploring swarm behaviors through crowdsourcing. So here what we have on the right are scenarios, and on the left we have different nanoparticles editors, so we can go, the crowd can go and design different nanoparticles, click inject, and see those particles and their collective behavior in our virtual tumors. And this actually uses parameters from our nanoparticle simulator that are relevant. So we taught the crowd lots of things. We taught them about tumor cells and healthy cells, because otherwise the solution to everything is to dump loads of drugs, and we wanted to be more specific with our treatments. We taught them about changing the dosage of the nanoparticles when you inject them, combining particles. You can change the size or the speed of the particles. Some of these cancer cells have molecules or receptors on their surface, and you can make particles that stick to those receptors. You can load those particles with a drug, and you can make a smart material where if something activates the particle, it's going to release its payload in the environment. And then finally, we have the ability of these particles to self-assemble and to disassemble, which speeds them up or slows them down. So if you're a swarm roboticist, you start saying, you know, these particles have an ability to sense their environment, to act on their environment by releasing something. That thing they release could activate another particle, so now you have communication. And through self-assembly and disassembly, you can change their motion. Even though I can't make them go left and right like I could with the flying robots, I can make them stop and go, or speed up and slow down. So this has led to around 180,000 simulations done by the crowd around the world, and we've since then started using this to train a neural network so that we could bootstrap and essentially give it a scenario, and this trained neural network could spit out the right vehicle design for that specific scenario. And we also spent some time playing our own game so that we could start to explore some of the more swarmy behaviors we were interested. So here is an example of trail formation, loosely inspired from the way ants do trail formation, but we've relaxed all directionality. They can't go left or right, for example. Turns out we've ended up with something that looks a lot like diffusion limited aggregation, if any of you are interested in this. And we started wondering, how could we actually test this in the tiny scales? How could we see these algorithms emerge? And we thought that one way to do it, rather than have to engineer chemically a specific particle, might be to give them a sense of augmented reality. And so this is the idea. We have real particles that are micro or nanoscaled, and rather than engineer their ability to communicate, for example, we could project light halos around them. And that would allow you to test with kind of a merge between the real world and the simulated world, how this influences the collective behavior. So you could change their communication range very easily, for example, and you could have particles that react to that light that's projected by doing something interesting. So in the case of trail formation, you might, for example, project a light halo. These particles would then stick to this light halo, and you would detect that. And then you would project a communication halo around those stopped particles, and maybe you could do something like trail formation in the real world at the tiny scale. And so with this in mind, we've designed the dome, which is essentially our system for closed loop control of micro agents. Essentially what it is a projector, and this projector projects in around 800 by 400 pixels. Each pixel is the size of a cell, depending on your magnification. And then we can detect using a Raspberry Pi camera what's happened and change our projection. So that allows us to do things like augmented reality. Here you see microparticles receiving communication halos and forming a trail. And here you see game of life that's virtually imposed upon this biological sample. When here all these bubbles, the blue ones, that are floating are volvox. And those volvox, so the natural world is seeding the artificial world. And here we're running the game of life rules in honor to Conway, who sadly died of COVID as part of this crisis. So when you go from the robot world to the nano world, you start thinking in huge numbers. And so since then, we've started wondering what we could do, you know, in things that worked in the tens of thousands, hundreds of thousands, maybe the millions. And so this is just an example. If you think of these farms, you know, if you were to plant seeds, you would sequentially zigzag through the farm and plant seeds one after the other in a sequential way. But if you think in the same way as the nano and the micro world think, well, maybe you would have 100 million robot seeds that deploy over this farm field, plant themselves, and biodegrade with maybe just the right amount of nutrients that they need for that farm. And it turns out it's not that crazy to think in huge scales with very simple rules that are inspired from the micro and nano world. So you remember that trail formation I showed that was in the simulated nano world? Well, this is that same rule set on a swarm of 100 coin-sized real robots in the laboratory. And just simple random motion, local communication, and you see they go from random motion to the emergence of these trails with very little calibration. So you could see how you could, actually there's no calibration, you could see how you would scale this up just producing robots and putting them in the scenario. Here what you're seeing is decision making with a swarm of 400 robots. Here the robots, this is inspired from honeybees, but again, random motion, really simple local interactions. And the goal is for them to turn blue if they're making the right decision. And here they go. They're blue from the initial state. It was blue and red. And here's an example of morphogenesis inspired from how shapes form during embryogenesis. So here our robots, we have 350, are essentially serving as cells. And they have two morphogens that they react locally on the robot. And then these morphogen concentrations virtually diffuse throughout the swarm. And what you get is spots and stripes, very similar to what Turing described you would get with cellular systems. And we can use those spots to grow artificial limbs and create self-organized shapes. And because of this self-organization, you can chop the limbs off, you can split the swarm and they regrow. So we get a lot of features for free from these very, very basic fundamental behaviors that actually would work at the micro and nano scale and work in robotics when we scale up to huge numbers. So the last thing that we're trying to do in my team is to figure out how to make swarms for people, because I feel like we've cracked a little bit the hardware, a little bit the software, And so we're trying to make this a reality. And so one of the papers I'll be presenting at this conference has to do with a use case study we did with firefighters, with people who work in warehouses, with people who do bridge inspection, to understand what they would care about in the deployment of swarms in their workplaces. And it turns out they're interested, they're keen, but we need to do it right, not a surprise. And we do a lot of public outreach as well. So this is Swarm Escape. And a proper escape route takes you between 30 minutes to an hour to break out, where you learn about swarm robotics while having fun. So thank you very much to everyone who's worked with me. This is very cross-disciplinary, and so there's a lot of different partners who have worked with me on all of this. And if you have any questions, I'd be happy to take them.